Okay, welcome back everyone. So in a previous video, we spoke about the neuromuscular junction. We said that there's a motor neuron that sends an action potential down the axon, down to the axon terminal, to the synaptic end bulb. We said the synaptic end bulb contains vesicles that contain a neurotransmitter called acetylcholine. We said that the acetylcholine is released through a method known as exocytosis only because calcium influxed into that nerve cell to tell acetylcholine to be released into the synaptic cleft. Finally, it binds to the motor end plate, to these acetylcholine receptors, which really are sodium channels. So sodium rushes in, creates excitation or an action potential that moves down the T tubule. The transverse tubule butts up against the sarcoplasmic reticulum where calcium is stored. And calcium is going to do something magical to the sarcomere. The sarcomere is where we have thick and thin filaments. The thin is called actin, and the thick is called myosin. Something is happening with the calcium to make muscles contract. So we have contractile proteins called actin and myosin. The one on the left here is myosin, and this one here is called actin. Now, if we look at actin, actin is the yellow one or the mustard colored one. And you'll see that actin have these round little circles. And each circle has a binding site attached to it. And it's called a myosin binding site, right? These little black circles. You can't see the full thing because there's another type of protein called tropomyosin, which is this string. This cord or this rope that we see here is covering these binding sites that we find on actin. And what's holding that string or that rope in place is another type of protein called troponin. Those are those blue little staples. Think of troponin as a staple holding down the string. Okay. On the left, we have myosin, and myosin has myosin heads. And what needs to happen is that the myosin head, this needs to make contact with those binding sites on actin. Okay? You'll see that on the myosin, it's called an actin binding site because myosin head has to connect with the binding site on actin. Make sense? So the actin binding site has to articulate with the myosin binding site. Where is the myosin binding site? On actin. Where is the actin binding site? On myosin. Okay. So actin and myosin, these are contractile proteins, whereas troponin and tropomyosin, these are regulatory proteins. Regulatory meaning it's the on and off switch. So troponin and tropomyosin being the rope, and troponin being that staple that's holding the rope in place, covering the binding site, it has a really important job because the only time that myosin can connect to actin is if the binding sites are exposed. And the only way they can be exposed is if these staples are removed. If we go in and remove troponin, which is the staple, and we move all of these staples out, guess what happens to tropomyosin, which is the rope-like structure that's covering the binding sites? Well, if the staples are removed and that rope or thread shifts out of the way, then all of the binding sites are exposed, and now myosin can attach to actin. And that's when contractions take place. So the question we have to ask ourselves is, well, that's pretty cool, but what is it that can remove troponin? What is it that can do that? Well, the magic key that can do that is called, once again, calcium. So calcium comes in, goes after troponin, removes it, tropomyosin shifts, and now, like a magnet, the myosin head can attach to the actin, and when they connect, a contraction takes place. The structural proteins are titin, nebulin, alpha, actin, myosin, and dystrophin. These deal with like the alignment of the muscle, um, the elasticity and flexibility of muscles. These are the structural proteins. And you've heard of people that have autoimmune issues against dystrophin. We just call it muscular dystrophy. That's when there's an autoimmune attack against this structural protein. So on the top here, we're looking at the orientation of what actin and myosin look like, right? Myosin, which is the thicker one in the middle with all of the myosin heads, and actin is thin, right? So the thin one is actin, and the thick one is myosin on the left. And you could see in this particular case, right, all those spheres you could see here, they have these binding sites. Those holes are the binding sites, but you can see the holes are covered by tropomyosin. And tropomyosin is held in place by the staple called troponin. Now, if calcium comes in, and goes after troponin and plucks those out, think of calcium as the staple remover. If it plucks out troponin, then tropomyosin shifts. And when that happens, the binding sites on myosin can attach. And when they connect, and these connect above and they connect below, we call this a cross bridge that forms. We call it a cross bridge. And when they connect, they create a contraction. They move it closer together. This moves this way, and this will move this way. Okay? So the myofibrils are composed of microfilaments. Some are thin and some are thick. The basic contractile units are called a sarcomere. Right? So the sarcomere is composed of three things: the contractile proteins, actin and myosin, the regulatory proteins, tropomyosin and troponin, those are the on and off switch, and then the structural proteins, titin, dystrophin, nebulin, myosin, connectin, and they deal with the alignment, stabilization, elasticity, and extensibility of muscle. So here's a close-up view of our actin, and these right here, those are the binding sites. So what needs to attach to it? Let's say this is the myosin heads, right? Let's say this is myosin. This myosin head right here is trying to make contact with that, but tropomyosin is covering it. So if calcium comes in and calcium goes after troponin and removes these yellow staples, then guess what happens to tropomyosin? They shift out of the way, they move, they translocate, and if this moves down and away, then guess what becomes available and exposed? The binding sites. And then these two, like magnetism, they meet and a contraction takes place. This is what the myosin heads look like, but there's really, so many of these myosin heads. Think of it as like a tug of war, right? If you have both hands on a rope during a tug of war and you pull the rope into you, eventually your hand needs to release and grab in front to grab the rope again to pull back. And then the other hand releases and goes in front of the one that's still contacting the rope. And then you pull. And then it has to release again to grab the rope on an available slot. So the hands moving, grabbing the rope would be the myosin heads. The exact point of attachment on the rope would be the binding site. Okay? So let's kind of review what's happening again. So the steps that initiate a contraction on the left and on the right would be the steps that end the contraction. So we have the motor neuron. Here is the synaptic terminal or the synaptic end bulb. And what's stored in there, remember those synaptic vesicles that contain acetylcholine. So calcium has to rush in to the synaptic end bulb. It tells the acetylcholine to release in the synaptic cleft. And when the acetylcholine is released, it's going to bind to the receptor. Those receptors open up after acetylcholine binds to it, and sodium, Na, sodium rushes into the cell. When sodium rushes in, it goes down the T tubule, the transverse tubule. The transverse tubule butts up against the sarcoplasmic reticulum. That's where they contact, right there. The sarcoplasmic reticulum stores calcium, so it's going to release the calcium. The calcium that it releases is going to hit the sarcomere, where you have actin and myosin. This calcium is going to go after the regulatory protein. It's going to go after troponin. It's going to remove troponin, so tropomyosin shifts, and then we have active site exposure. When you have the active site exposure, myosin connects to actin, creating that cross bridge formation. And a contraction begins. And it moves this way, and those myosin heads attach, moving actin that way, right? So this is myosin, and the red here, that's actin. Now, if you want the contraction to end, well, we have to get rid of acetylcholine. So acetylcholine is removed by an enzyme called acetylcholine esterase. The E is for esterase. Acetylcholine esterase. And remember, anything that ends in ase is an enzyme. So you have acetylcholine esterase that's going to degrade and break down the acetylcholine. If acetylcholine is broken down, then there's nothing to open up that sodium channel to allow excitation to hit the sarcoplasmic reticulum to release calcium. So if you don't have the release of calcium, then the sarcoplasmic reticulum recaptures it and puts it back in the storage container. If the calcium is back in its container in the sarcoplasmic reticulum, that means it's not going after the regulatory proteins of troponin, which means the binding sites are exposed. So if those binding sites and the active sites are now covered, then no cross bridge forms, contraction ends, and the muscle relaxes. Okay? One of the things that I want to point out is that in order for the myosin head to detach right here, we need ATP. We need adenosine triphosphate in order for the detachment to take place. It takes energy to release the myosin head. Okay, so on the left, right, everything here on the left side goes along with everything here on the left side. Okay, so when you watch this back, everything here on steps that end contraction go along with everything here on the right-hand side. Okay, so I'll keep you there just for a second. You can pause and then you can work your way through this, but this just repeats all the steps that I just went through with you several times. The one thing that I pointed out at the end was that the contraction begins as repeated cycles of cross-bridge binding, pivoting, and then detachment. But what I said was that the detachment occurs by using ATP. In order for the myosin head to detach, you need ATP. Okay, again, I'll review it. The neuromuscular junction, acetylcholine is released by the synaptic end bulb. And when that acetylcholine is released, it's going to alter the action potential, the transmembrane potential, creating an action potential that spreads right into the T-tubule. The T-tubule butts up against the sarcoplasmic reticulum, and the sarcoplasmic reticulum is going to release calcium. When it releases calcium, calcium binds the troponin. It's going to remove troponin, creating a shift in that troponin tropomyosin complex, exposing the active sites so that myosin head can actually bind to it. Okay. When the contraction ends, we said acetylcholine is broken down by acetylcholine esterase. The sarcoplasmic reabsorbs the calcium, it goes back into storage, and then when there's no more calcium, the troponin and tropomyosin returns to its normal resting position where the binding sites are covered, and at which point there's no cross-bridge interaction. So a sliding filament theory or a contraction cannot take place. And the muscle relaxes. It goes back to its resting state. Okay, when I come back, I'm going to talk, actually, I'm just going to finish this slide. It's only 2 slides, and this kind of goes along. So myosin is going to pull on actin. And when it does, it causes the thin filament. We know the thin filament is actin, right? It's going to move it to slide inward. And thanks to the structural proteins that I mentioned before, all of those structural proteins here, thanks to those, there is transmission of force throughout the entire muscle, resulting in a whole muscle contraction. So if we look at the top left here, we look at like the 12 o'clock position, you have actin, and we see that the binding sites on actin are exposed. Okay, we see here troponin, we see tropomyosin, but it's not covering the binding site. So what happens to myosin? See, myosin starts to reach out and says, hey, we've got exposure here. We're ready to connect. But what do we have hanging out here in the wind, right? Right here, we have ADP and P. So ADP, remember, we start off with adenosine monophosphate. What does mono mean? Mono means one. If we add another phosphate to that, right? If we add another phosphate, now it's going to equal adenosine diphosphate. You have ATP. Now, if we add another phosphate to ADP, right? ADP is 2 phosphates, adenosine diphosphate. Now, if we add another phosphate to ADP, then we're going to get ATP, adenosine triphosphate. So right here, we see we have adenosine phosphate, adenosine diphosphate, with another P, another phosphate hanging out in the wind. Typically, that phosphate doesn't hang out by itself. It attaches to something called creatine. Creatine. So creatine and phosphate attach, so it's called CP, creatine phosphate. Okay, phosphates just don't hang out by itself, it has to attach to creatine. So it's called creatine phosphate. Okay, so now look what happens between step 1 and step 2. Now they bind. See, there's a cross bridge that forms. Now we can see the myosin head attaching to the binding sites. Now look what happens to this ADP and P right here. Something is starting to happen with that phosphate, but look at the myosin head. Let me erase it here. Right here. Okay. Look at the position here of myosin and then look at the position here. See how this one has a kinked angle? And here it doesn't have that kinked angle yet. So here it made contact, and here, see the arrows right here? It flicked it. It flicked it. It's creating a slide. It's contracting. It's moving it. Now, after it slides, we want this to detach to articulate again. So that ADP and that other creatine phosphate combine, see it here? The ADP and that other phosphate combine to form ATP, and now with ATP, it can detach. It needs energy to detach. And we know that because you know what? When someone, let's say in CSI Miami or CSI New York or crime scene investigation, whenever they find a dead body, sometimes they're in a state of rigor mortis. Rigor mortis is the person is dead and they're in a flexed, shortened, contracted state. Why? Because the muscles couldn't release. Well, why couldn't they release and relax? Well, they're dead. Yeah, but when they're dead, what can't they do? They can't make ATP. If you can't make ATP, those proteins are still attached. They stay in a short and contracted state, at least in the early stage of a deceased person. Now, after hours and days, the body does release digestive enzymes to break down all the proteins, so eventually it does release. Okay, so that's a little bit about the contraction cycle. When we come back, we're going to talk about an interesting topic that is typically a hot topic around Thanksgiving time if you celebrate with turkey. And that concept is, do you want dark meat or white meat? Do you prefer the leg or the breast? When we come back, we'll get into that topic.